Our next speaker is Margaret Feeney, who's joining us today from UCSF. And Margaret's focus is on the infection and developing humans with and one particular interest is in the acquisition of immunity to malaria in infants and mothers. Welcome.
Great, thank you so much. It's been a wonderful session so far. Let's see, here we go. So malaria in pregnancy continues to be an enormous global problem, impacting 1 out of 3 pregnancies in Africa. And every year, a huge number of pregnancies occur worldwide in regions that are at risk for malaria transmission. Furthermore, it's been shown in several observational studies that infants who are born to mothers with malaria during pregnancy may themselves have altered susceptibility to childhood malaria. The clinical consequences of placental malaria are substantial. They include maternal anemia, as well as intrauterine growth retardation, low birth weight, prematurity, and even sometimes miscarriage and stillbirth. And as a result, placental malaria is estimated to account for about 100,000 infant deaths per year. Placental malaria occurs when infected red blood cells accumulate in the placenta and compromise its function. It is believed that an important mediator of this is VAR2SA, which we heard about earlier. It mediates adherence to chondroitin sulfate A in the placenta and is now the subject of a major vaccine effort. I'll just point out that prima gravidas are at highest risk in areas of high transmission of malaria, because subsequent to the first pregnancy, antibodies to VAR2CSA are frequently acquired, and these provide some relative protection against placental malaria in future pregnancies. So compared to normal placental histology, which is shown in the top, we see sequestration of infected red blood cells in the microvasculature, as well as infiltration of monocytes and macrophages with variable degrees of inflammation. There is breakdown of the syncytial trophoblast barrier, which can become just completely denuded in some areas. And there's also deposition of hemozoin and other red blood cell breakdown products. And these processes ultimately result in placental insufficiency and poor fetal growth, although the exact mechanisms by which this occurs are not yet fully understood. But what I want to talk about today is not so much the parasites or what happens in the moms or in the placentas, but rather what happens on the other side in the baby, and specifically what happens with the fetal immune system. My lab's been interested in the question of whether and how the infant immune system sees malaria antigens during gestation and how they respond to these antigens. So when considering this question, it's sort of important to couch it against the timeline of fetal development because the fetus's immune capabilities are rapidly changing from week to week and month to month. And in any individual case of placental malaria, it's very, very difficult to tell exactly when the infection occurred because most of these infections are clinically silent and we don't have a good non-invasive window into the placenta. But the placenta can become infected as early as 8 or 9 weeks of gestation, which is when blood first enters the intervillous spaces of the placenta. And increasing data shows that in areas of high transmission, the bulk of these infections are probably established late in the first trimester or early in the second trimester. And this is very important because at this time, early fetal T cells are just beginning to populate the periphery. And these early and mid-gestation fetal T cells are transcriptionally and functionally distinct from those that are seen in later life. I could give a whole nother talk on how they differ functionally, but just in terms of why. Successful pregnancy requires reciprocal tolerance between the mother and the infant, and numerous mechanisms have evolved in order to enforce this tolerogenic in utero environment. These include factors that are T-cell intrinsic and largely epigenetically governed, like suppression of the interferon gamma and other Th1-linked loci, but also T-cell extrinsic factors such as a suppressive cytokine milieu and immaturity in antigen presentation machinery. And these redundant forces sort of work together as brakes to restrain any fetal inflammatory response to antigens that are encountered in utero. Otherwise, this could trigger parturition and threaten the pregnancy. So what then happens when the fetus does see non-self antigens? I think this has been best studied in the case of maternal alloantigens, where it's been shown that at mid-gestation, fetal T cells have an intrinsic propensity to differentiate into FOXP3 positive T-regs that become functionally suppressive upon encounter with non-self antigens. And this is probably crucial so that the fetus does not mount an inflammatory assault on maternal cells and tissues. But this raises the obvious question of how does the fetus then respond to pathogen-derived antigens like malaria if they are encountered in utero? So to answer this question, my lab has been using cord blood samples from the PROMOTE study, which is a randomized controlled trial of malaria chemoprevention in Uganda. In this study, we enrolled 300 pregnant women at 12 to 20 weeks gestation, and we randomized them to receive SP per WHO guidelines or DP either monthly or every 3 months. And then participants were monitored throughout pregnancy with both active and passive surveillance for malaria. And when the babies were born, we collected the placentas, and we also collected large volumes of cord blood, actually into blood donation bags, so we got a lot of cord blood. And the clinical outcomes of this trial were published a few years back, showing that DP, when given monthly, was very highly effective at preventing malaria in pregnancy and placental malaria. Interestingly, however, it had no impact on infant birth weight. So in cord blood, the vast majority of T cells are naive in phenotype. However, we found that in malaria-exposed infants, there were higher frequencies of effector-differentiated memory CD4 cells. And this was particularly apparent in those who had active placental infection at the time of birth. These CD4 cells frequently express Ki67, which is a marker of in vivo proliferation. So this suggests that in the setting of placental malaria, fetal T cells expand and differentiate in vivo. And next, we assess the functional capabilities of these T cells. Upon stimulation, placental malaria-exposed infants had significantly higher production of interferon gamma and TNF. This was most evident in the CD4 T cell population, but it was also seen to a lesser and more variable extent among CD8 T cells. And we wanted to know if these cells were malaria-specific. So we measured the proliferative response to a schizont extract and found that malaria-exposed infants had significantly higher proliferation of both CD4 and CD8 T cells, and this could be abrogated by antibodies blocking MHC1 and 2, respectively, indicating that this is a conventionally MHC-restricted TCR-mediated response. So taken together, we thought this was very good evidence that malaria-specific CD4 and CD8 T cells are indeed primed in utero. But because schizont extract, sort of a messy gamish of an antigen, we also tested proliferation response to a pool of overlapping peptides spanning MSP1, which is a blood stage antigen. This gave us very clean data with very minimal background. And again, we saw proliferation of both CD4 and CD8 cells in response to the MSP1 peptide pools, but this was only seen in infants who had had placental malaria. So what about T-regs? Our original hypothesis had been that T-regs would be induced in utero by in utero malaria exposure, sort of in parallel to what we had seen with maternal alloantigens. But in short, we didn't see much of a difference. We did see somewhat higher T-regs amongst babies whose mothers were already parasitemic at the time that they were enrolled in the study, possibly indicating early pregnancy exposure to malaria. But when we compared infants based on placental histology or placental lamp at the time of delivery, we saw essentially no difference. An interesting twist, though, is that we found male infants had significantly more cord blood T-regs than females. And when we stratified this by placental malaria exposure, this difference was mostly driven by the malaria-exposed infants. And this prompted us to look more broadly at sex differences in other immune parameters. Interestingly, sort of the pattern that we see really perfectly echoes the pattern that has been widely reported in adults, wherein male infants had more naive and regulatory T cells, whereas female infants had more activated and differentiated cells, as well as higher levels of IP10, which is also known as CXCL10. It's an interferon gamma-driven chemokine that drives effector T cell differentiation. And just for anyone in the audience who's not familiar with this really fascinating and growing literature on sex differences in the immune response, I would just call attention to this paper that was published a few years ago by Sabra Klein here at Hopkins, in which they did a really interesting retrospective study of retrospective analysis of data from phase 3 trials of RTSS, in which they found higher all-cause mortality in female vaccine recipients, as well as a trend towards a higher risk of fatal malaria in girls who had received RTSS. And there are other hints, I think, over the past few years since this was published, of sex differences in the immune response to malaria. But it's completely fascinating to me that this sexual dimorphism actually can begin in utero very early on. And I think this is also an important point to consider for translational purposes when we look at vaccine trials in infancy. So we next asked whether the malaria-specific CD4 T cell response that we observed in cord blood would correlate with an infant's risk of malaria following birth. And because the trial design from which we got these samples included a second phase of randomization in which infants were randomized to postnatal chemoprevention from birth to age 2, we restricted our analysis to the kids who were in the every 3-month DP arm, because we figured that the kids who were getting monthly DP as directly observed therapy would just not be getting any malaria at all. And we performed 3 separate outcome analyses, looking first at malaria incidence, at the risk of parasitemia, and then a proportional hazards analysis of the time to first malaria episode. And we found that for each of these 3 outcomes, having higher malaria-specific CD4 proliferation was associated with protection, including protection from parasitemia as well as clinical malaria. And this remained true whether or not we adjusted for clinical variables to the extent that we could, which included gravidity, maternal malaria during pregnancy, and maternal chemoprophylaxis assignment. So having this evidence that the fetus mounts T cell responses to malaria antigens in utero, we next sort of pivoted to the question of how are these antigens entering the fetal circulation. And just a little primer on placental function for people who don't think about placentas every day. But the placenta is actually a remarkably effective biophysical barrier. It allows for the transport of gases and nutrients and fluids, things that the baby needs. But generally, it excludes cells. So there are very, very, very small number of cells that get across and account for the microchimerism, which is a very low percentage. But by and large, hematopoietic cells of the mother are completely separate from those of the fetus. So what does get across? IgG does. This is via an active transport mechanism such that at full term, infants generally have IgG levels that exceed those of their mother. But this process is selective, as I'll come back to in a few slides. In terms of microorganisms, some microorganisms can and are famous for causing congenital infections, but P. falciparum, by and large, is not one of these. There are very rare cases reported of congenital malaria in which intact parasites cross over to the fetal circulation and develop a self-propagating blood stage infection. But these are believed to be due to microtransfusions, which are sort of like, you can think of as hemorrhages from the mother into the baby. That is not the normal outcome of garden variety placental malaria. But the extent to which antigens derived from maternal pathogens cross over into the placenta is not something that is very well understood or been systematically studied. So we hypothesize that malaria antigens might be entering the fetal circulation by one of two routes. First, the inflammation of placental malaria, as I mentioned, can cause the syncytial trophoblast barrier to become denuded in some areas, which might just allow antigens to sort of pass freely. And this syncytial trophoblast layer is a single layer of fused multinucleated cells, which lines this tree-like structure called the chorionic villus. In the second half of pregnancy, this single cell layer is the only thing that's standing between maternal blood, which is filling up these inner villus spaces, and the villus core, which is where the fetal endothelium and fetal macrophages lie. But alternatively, if we zoom in here, malaria antigens might actually be ferried across the syncytial trophoblast in the form of IgG complexes by FCRN, which is also known as the neonatal FC receptor. FCRN is a pH-dependent shuttle that lives in the endosome and is responsible for the active transport of IgG into the fetus. So we wanted to know if FCRN might be acting as a Trojan horse and sort of shuttling malaria antigens into the fetus complexed to maternal IgG. And to answer this question, we designed an experiment to purify IgG from cord blood of Ugandan infants and then analyze the eluted proteins and peptides by mass spectrometry. And then we created a lookup database comprised of publicly available P. falciparum proteome information and supplemented this with sequences from multi-gene families that were obtained at various points from around the globe. And then we compared the peptides that we identified by mass spec to this query database, enabling us to look in an unbiased and sort of proteome-wide fashion rather than focusing on candidate antigens or the usual suspects. And these data are very, very preliminary. This is kind of hot off the press and just coming in. But what we found, I think, is quite striking. We found that there was abundant IgG complexed P. falciparum antigens in the cord blood at the time of birth. When we looked in a blinded fashion, nearly all of the infants with placental malaria had malaria-derived peptides in their blood, usually multiple different peptides and proteins, whereas none of the 20 US-born controls had any hits. And in fact, the number of proteins that we detected correlated with the diffuseness of the placental pathology that we had seen. And the peptide sequences overwhelmingly mapped to sequences from East Africa in the variable gene. So that gave us more confidence that this was a very specific signal that we were detecting. So I mentioned that FCRN transfer is selective. It only transports IgG. It doesn't transport the other isotypes, but also the transfer is very influenced by FC region characteristics of the antibody, including the subclass and glycosylation as well. In terms of subclass, IgG1 is preferentially transported. And in most studies, it's been shown that IgG4 is the next most abundant in cord blood, followed by IgG3 and IgG2. However, I would just point out that there is a polymorphism that's been described in IgG3 that's rare in Caucasians, but actually pretty common in Africa, which increases dramatically its binding to FCRN such that it's transferred almost as efficiently as IgG1 is. And then the other factor that highly influences FCRN transport is the N-glycosylation state of the antibody. I think we're really sort of only at the dawn of understanding this, but I expect it's going to be very important for those of us who are interested in maternal vaccination as a route towards protection of infants. Just to give you a flavor, it's been shown that antibodies that are digalactosylated are transported much more efficiently than those that are non-galactosylated. And fucosylation and sialylation have also been reported to influence transfer via FCRN. So this selectivity in IgG transfer has important functional consequences because these subclasses differ in their antimicrobial effector functions. And in particular, IgG1 and IgG3 are together considered the ones that are cytophilic antibodies because they are able to bind the FC receptors on many immune cells. And they're also able to bind complement and opsonize for phagocytosis. IgG3 is the subtype that does this the most efficiently. And I think it's relevant that malaria-specific antibodies of the IgG3 subclass have been shown in several studies to be associated with protection from malaria. And intriguingly, Christopher King's group published this study in which the IgG3 polymorphism that I mentioned earlier that increases binding was studied in a large cohort of infants in Benin. And it was shown that those who had this polymorphism had higher IgG3 levels, malaria-specific IgG3 in cord blood, as well as a longer half-life of IgG3, because FCRN also influences recycling of IgG. And this translated to protection from malaria during the first year of life. So it seems that these cytophilic antibodies do something good for the babies. What is it that they do? The answer is probably more than one thing, but I would just propose that one important role may be that the maternal origin IgG that gets across and is able to bind FC receptors on innate lymphocytes provides a means by which these infants' innate cytotoxic effector cells may respond to a specific stimulus. So in effect, it's like the baby is able to borrow a specific antigen receptor from the mother at a time when they themselves don't have any adaptive immunity or immune memory. And indeed, we have seen evidence of activation in utero of both CD16 positive fetal NK cells, as well as the V delta 2 subset of gamma delta T cells, which in some instances upregulate CD16. These gamma delta T cells are of particular interest in my lab because they have been shown previously to have intrinsic reactivity to malaria via this semi-invariant TCR that it expresses, which recognizes phosphoantigens that are produced by the plasmodial apicoplast. But in some instances, and particularly in infection, they upregulate CD16 as well. And when they do, they are able to be directly activated by opsonized red blood cells, as we showed in this recent paper. So the V delta 2 gamma delta T cells have two different ways that they can work as anti-parasite effectors. One is TCR mediated, sensing phosphoantigens. The other is via CD16, which is an activating FC receptor, which can be triggered by opsonized parasites directly. So that makes it sort of a multifunctional effector cell that can contribute to immune surveillance. So just to conclude, fetal T cells do see malaria antigens before birth, and malaria-specific T cells are primed in utero. The fetus is capable of mounting functional Th1 polarized CD4 and CD8 T cell responses. Importantly, the immunity is sexually dimorphic, even in utero. And IgG-bound malaria antigens are actively transported into the fetal circulation, which may ultimately have some role in priming CD4 and CD8 T cells, but is also relevant in terms of activating FC receptor-bearing innate fetal lymphocytes. I just want to acknowledge my group, including all the people in my lab who've worked on these studies, as well as our fantastic collaborative group who did all of the clinical work, and as well as some of the lab work in Uganda. Thank you. Happy to answer questions.
Let me ask you this, the FC, remind me, you'll have to repeat it.
Okay, I can.
Oh, I'm sorry, I thought it was coming down.
Do you want me to?
Oh. Well, here, I could just, can I borrow your microphone for a second?
Sure.
Can you remind me, the FCRN is present on tissues other than the placenta?
Yes.
And has but is your sense is that it may have evolved to do exactly what you're describing? And I was curious about the glycosylation, need for glycosylation. Does that change at all during pregnancy? Is there an alteration in glycosylation that might make it more effective or sort of divert things to?
Yeah. Great question. So the first question was about FCRN, whether it evolved for this fetal transport. I can't comment on its evolutionary purpose, but it does. That's one of its major purposes. But another very important purpose is in IgG recycling. So it very much affects the half-life and is now very much an industry target in order to sort of increase the longevity of monoclonal antibody therapeutics. So it is not only relevant in the placenta, it's expressed pretty widely. Second question was glycosylation. So there have been a few studies whether glycosylation, so glycosylation is very influenced by inflammation, for instance. And there have been a couple of studies showing that it is altered during pregnancy, but they conflict. The data conflict. And I think that the field is kind of young enough that we're still just getting a handle on it, but it would make sense maybe that during the state of pregnancy, there might be some alteration of this that evolved to influence what the baby was getting in terms of passive immunity. Yeah, thanks.
Great, thanks so much.
Thank you.